Erythrocyte, red blood corpuscle, red blood cells, RBCs, are the non-nucleated. Red blood cells are also known as erythrocytes. Erythros equals red. Red color of the red blood cell is due to the presence of the coloring pigment called hemoglobin. RBCs play a vital role in transport of respiratory gases. RBCs are larger in number compared to the other two blood cells, namely white blood cells and platelets. RBC count ranges between 4 and 5.5 million CUMM of blood. In adult males, it is 5 million CUMM, and in adult females, it is 4.5 million CUMM. Normally, the RBCs are disc-shaped and biconcave, dumbbell-shaped. Central portion is thinner and periphery is thicker. The biconcave contour of RBCs has some mechanical and functional advantages. Red blood cells are non-nucleate. Only mammal, which has nucleated RBC, is camel. Because of the absence of nucleus in human RBC, the DNA is also absent. Other organelles such as mitochondria and Golgi apparatus also are absent in RBC. RBC has a special type of cytoskeleton, which is made up of actin and spectrin. Average lifespan of RBC is about 120 days. After the lifetime, the senile, old, RBCs are destroyed in reticuloendothelial system. Functions of red blood cells. Major function of RBCs is the transport of respiratory gases. 1. Transport of oxygen from the lungs to the tissues. Hemoglobin in RBC combines with oxygen to form oxyhemoglobin. About 97% of oxygen is transported in blood in the form of oxyhemoglobin. Chapter 125. 2. Transport of carbon dioxide from the tissues to the lungs. Hemoglobin combines with carbon dioxide and form carbhemoglobin. About 30% of carbon dioxide is transported in this form. 3. Buffering action in blood. Hemoglobin functions as a good buffer. 4. In blood group determination, RBCs carry the blood group antigens like A antigen, B antigen and Rh factor. This helps in determination of blood group variations in number of red blood cells. Physiological variations. Increase in RBC count. Increase in the RBC count is known as polycythemia. 1. Age. At birth, the RBC count is 8 to 10 million CUMM of blood. 2. Sex. Before puberty and after menopause in females, the RBC count is similar to that in males. During reproductive period of females, the count is less than that of males, 4.5 million CUMM. 4. Muscular exercise. There is a temporary increase in RBC count after exercise. 5. Emotional conditions. RBC count increases during the emotional conditions. 6. Increased environmental temperature. Increase in atmospheric temperature increases RBC count. Generally, increased temperature increases all the activities in the body including production of RBCs. 7. After meals, there is a slight increase in the RBC count after taking meals. It is because of need for more oxygen for metabolic activities. HAEMOPOIESIS. It is the development of blood cells, i.e. RBCs, WBCs and platelets. Therefore, the term hemopoiesis includes erythropoiesis, i.e. development of RBCs, 2. Leukopoiesis, i.e. development of WBCs, and 3. Megakaryocytopoiesis, i.e. development of platelets. Variations in size, shape and structure of RBC. 1. Anisocytosis, variation in the size of RBCs. 2. Poikilocytosis, variation in the shape of RBCs. 3. Spherocytosis, spherical RBCs, more fragile. 4. Anemia, Reduction in number of RBCs less than 4 million per microliter, or their content of hemoglobin less than 12 gm, DL or both. 5. Polycythemia, RBC count increases more than 6 million, micro-L. Causes: A.